let's talk today in the third lecture of the series Regulatory Science and Translational Research about the non-clinical development of gene therapies and cell-based therapies. And that is almost a paradigm of translational research, as you will see, because we have a lot of new formats of medicines where the same principles apply, even if they're not gene therapies and cell therapies. Excuse me, just short introduction of about myself. So I'm currently, that we are all online, we need to almost say where we are. So I'm located in the north of Denmark. You can see this here on the map in a town called Aalborg. So I have the pleasure now to being able to work from home. I recently joined Pharmalex, which is a consultancy company, as head of biopharma excellence and chief medical officer for the biopharma area. Before that, and it was until end of summer this year, I was chief scientific officer at the MHRA. That's the Medicines and Healthcare Products Regulatory Agency in the UK. And I was director of one of their centers, the National Institute for Biological Standards and Control. And as you can see here, I will not go through all of these details. I also saw two other agencies before. So I was the medical head of the licensing division for medicines at the Danish Medicines Agency and head of a division at the Paul Ehrlich Institute, where I worked for 8 years. And at the European Medicines Agency, I was delegate for first for Germany and then for Denmark. So I was member of the main committee, the Committee for Medicinal Products for Human Use, and I was chair of the Committee for Advanced Therapies, which is maybe the most relevant for the presentation today. Now, what I'm talking today about very briefly is an overview of the requirements. So non-clinical, as you all know, this is basically, as the name says, anything that's not clinical. So that is usually the animal models, the animal testing of medicines, but this can also be in vitro tests or functional tests that are always also, sometimes also called non-clinical tests. And then I will talk about the principles. What is the mindset that we should have and that regulators should have, but also what developers and academic groups who work in that field towards a medicinal product should have. And as you will see, the best mindset, and I will spoiler this already now, is a knowledge and risk-based brains-on approach. And what that is, I will come to, and I'm using a focus group meeting, which we had at the Committee for Advanced Therapies quite some time ago, but the principles are still the same, are still valid. And I will also round this up with a few examples. So first, when somebody starts thinking about and reading about non-clinical guidance, there's a lot of documents out there. So the European Medicines Agency alone has quite a number of different reflection papers and guidelines, and also the US FDA has. So that can be quite a jungle. And then we do also have a checklist in the legislation, which is the Directive 2009-120. That is basically a directive which amends the Human Codex, as we call it. That's the European Medicines Law 2001/83 as amended. And there you can see already for gene therapy and for cell therapies that there's quite a number of different demands, requirements that companies need to fulfill or developers need to fulfill. And you can see already here that pharmacokinetics, that is the classical pharmacology in animals, is different. So for example, for gene therapy, this is not pharmacokinetics, this is biodistribution, because you inject it locally or you inject it ex vivo, so outside the human body. And then also the vector can be shed into the bloodstream, and that is also something to look at. So already here you can see that it is not all according to the classical principles. And so the list goes on. I will not go into detail here because we will hear about these principles a bit more hands-on later in the presentation. Now, how to do this in practice and what are the principles? You can lead perfectly confusing discussions about non-clinical development when you talk about different stages of development and also different disciplines. So, for example, the non-clinical development will have much bigger focus when it comes to the real translation of a medicine. That means from the animal models up into a first in human clinical trial where the medicine is applied to a human being for the first time, or later for marketing authorization when there's clinical data already, then of course it will be seen in a different light. It doesn't mean that it is less important, because in the non-clinical development, you can do things that you cannot do in humans. So for example, overdose or repeat dose if it is just a single dose application, like for a gene therapy, which is very often only a single dose application. And then by aim. So this is where sometimes the confusion comes from. Some people talk about the animal models as the proof of concept. So that is, it works, that is the efficacy. But there's much more to that. There's also the toxicology. It works safely. And not always will you find in a publication about a new gene therapy, for example, in an animal model, that it works safely. Well, there is, of course, a bit of that, but the study may focus more on the efficacy, that a gene defect can be corrected, for example. Now, how do we test that? So obviously we have to test it in a relevant species. And I'm saying that obviously because it is not always so obvious, but there's a definition in, now it is quite an old guideline already, the ICH S6 guideline, it's still valid. A relevant species is one in which the test material is pharmacologically active due to the expression of the receptor or an epitope in the case of a monoclonal antibody. Now, when it comes to cell therapies, there is not just one receptor or one epitope. There is many, many receptors on the cell surface. And there's also a lot of factors that the cells secrete and detect and recognize on other cells. So basically, you could say that the only relevant species for human medicines and human cell-based medicinal products is the human being. And of course, we can't do a non-clinical test in a human being unless we have a good approach, a so-called risk-based approach. We'll briefly touch upon this later. And there's also an own lecture on that. But for now, what is very often used is the homologous model, as we call it. So the classic now, since monoclonal antibodies are an established type of medicine now, is when you have, for example, a humanized or a human anti-human CD28 monoclonal antibody. So an antibody which has a human structure in itself, and it recognizes the human version of CD28, which is a signaling molecule on the surface of certain immune cells, then this will not work in animals. But you could engineer a rat format, anti-rat CD28 monoclonal antibody that will work perfectly in rats. And that is the homologous model. And in analogy to this, the same can be done, and sometimes not easy to do that, with human cells, like in this example, human engineered T cells with a human T cell receptor, mouse engineered T cells with a mouse T cell receptor are then the homologous model. And I will come back to this later in the presentation, because it is a very good model to show why it is a bit tricky sometimes to develop models like this. And we have had a lot of discussions in the regulatory world how to best test the medicine, the candidate medicine in animals. So obviously, as I said, advanced therapies, ATMPs, advanced therapy medicinal products, that's the abbreviation, are very complex medicines. So for cells, you could say the first possibility is to test the product in animals. The advantage is that you test the actual medicine, and you could then test all the impurities which are in there, and you might actually find a model where it's possible, at least in part. But as I said, the disadvantage is that not all interactions are similar to humans, or none at all, and then the model can simply not be used. And as I said, the possibility number two is the homologous model. The advantage is that all interactions are relevant. So pharmacology and toxicology you can all see. But the disadvantage again is it's not that actual medicinal product, and mouse is not man. So if you find a safety finding in mice, it might not be relevant for humans. And if you don't find anything, that doesn't mean that nothing will pop up in humans later. But at least this is the only thing we can do sometimes. And this is why we need to look so closely at the design of non-clinical models and non-clinical studies. And we had a number of questions at the time, because some colleagues said, well, then a homologous model should be used. But then others said, well, no, we want to have the medicine tested. And then even other ones said, well, can we find other approaches? Can, for example, in silico models, so computerized models work? And can we use existing models, existing knowledge? So for example, we have a certain type of immune cell that's engineered, and we have seen 3 similar ones already. They're not the same, but quite similar. Can we not use that knowledge? And then some people said, no, only the medicine itself is relevant, and anything else is, if at all, then only supportive. And what about very new animal models that are specifically developed for situations like this, but which may not yet be validated? And actually, what is the aim of these non-clinical tests? Of course, it is to detect toxicology, but if we already know some aspects, like for example, for embryonic stem cells, when they are differentiated into a tissue, we know that there might be some residual embryonic stem cells, which then can form a teratoma, as it is called, because that is what they do, they divide. So we know that already. We don't have to test that in an animal, because we know that this is going to happen if there is a residual a trace of embryonic stem cells. It is rather to estimate the risk of it. So in a thought process like this, it may actually be possible to circumvent such a model. Now, this is just very briefly these reports. So you can see this is a bit older. It's 10 years that we discussed that, but I kept this in the presentation because the findings are still relevant. The non-clinical discipline, also there are many new models available. The basic principles are still the same and will stay the same for quite some time. And we discussed with members from pharmaceutical industry and also from academic groups at the time. And the obvious outcome for one thing is, when it's individual products, better come for scientific advice. That means ask the authorities. It's typical for a regulator to say this is case by case. So this is very unsatisfactory, but sometimes the questions are so specific that it is better to come for scientific advice. And there is a lecture, an own lecture, about what scientific advice is and how it works, with a few tips on how what to expect and how to prepare for that. So the principles for following regulatory non-clinical guidance is: do not follow the guidelines for the sake of following guidelines. And that was a very important thing for the regulators to say at the time, because there are some other regulators in the world who still want to have, for example, a rodent and a non-rodent species for testing, and both may not be relevant at all. And that, of course, is, when it comes to the ethics of animal testing, in our mind, not a preferred approach, because you would basically not learn anything unless you know about the shortcomings and you can get a few data out of that which are relevant to inform the human study. Then, of course, it's a different thing, but that is something which one has to look at, of course. Then scrutinize the study design. How relevant is the outcome expected to be? And what will the shortcomings be? So when you know the shortcomings of the model, it's much better to put the data into context. And then obviously employ a risk-based approach. I will come back to this, and remember the 3R principles. That means the reduction and refinement of animal studies, so that the ethical aspect is also taken care of. And when we discussed with the stakeholders, they also said, well, you see so many different companies asking for scientific advice. So you see a lot of development programs. It's all confidential. Even that company is coming for scientific advice is confidential. But can you not explore the possibility of setting up a platform or some sort of a knowledge sharing? Because some questions will always be the same and the answers will always be the same. And that's true. And we did that, and I will show that. And then we also encouraged to use the regulatory incentives from early on. In another lecture, I already talked about the certification procedure for advanced therapies, which is the only data assessment procedure before marketing authorization. It's not been taken up very much in the European Union, unfortunately, but that can be used as a gap analysis as well, because you can have a look at non-clinical data as they exist and as they come in. And also innovation task force briefing meetings can be very useful in that. Now, what we at that time, or what the stakeholders at that time very much advocated, and we reflected on that and we accepted it, at least in part, was that there should be the possibility to rely, or at least be strongly informed by experience from similar products, and also, if available, previous clinical experience. For example, dermal products with scaffolds, or maybe chondrocyte products for the knee joint. We know a lot about these products. So the question is, in as much you really need to have a non-clinical study when you develop a new product, or where you can maybe make reference to another one. That is again case by case, but at least the possibility was discussed at the time, and I think is still considered. And the developers were very clear, and that was an important information for me at the time, that they didn't request a different standard, because sometimes the discussions were a bit, well, we're an advanced therapy, we're a small company, do we really have to follow all of that? It's too expensive. That never came up. It was rather that there should be recognition of the long-standing experience in some areas. Of course, most gene therapies are quite cutting edge and new, but there is of course the use of certain vector platforms where there might be authorizations already, or where there might be different clinical trials with the same vector but a different gene of interest inserted. Then of course it becomes interesting when it comes to reliance on certain data, and I think we will see quite a bit happening in this space. And then and that is again why I put it in the beginning, it depends on the stage of development. If there is already clinical data, then that may in part at least compensate for non-clinical studies. And a regulator should not ask for non-clinical study just because it's in the guideline when there is a clinical trial already there, unless there's a clear reason to study that in animals then. And then when it comes to what animals to choose, a lot of discussion on in vitro testing. So for example, cell systems, artificial organs, 3D matrices with cells inserted, that can actually be used. That has evolved quite a bit as a non-clinical model. And when it comes to these two different approaches, you remember proof of concept, it works. Toxicology, it works safely. Proof of concept is usually accepted to be a homologous model, so not the actual medicine, but something which really works. And you may actually see quite a bit of safety findings relevant. But for toxicology, there were some regulators, at least at the time, saying that it may also be necessary to test the actual medicinal product. And it could also be a disease model if available, because sometimes we would not see anything for proof of concept, it's obvious. But for toxicology, we may not see anything in a normal immune system in animals, or with a non-diseased animal. So it could be a disease model. But that, of course, is then becoming a very complex approach and would have to be discussed ideally with the regulatory authority before setting up such a model. And very interestingly at the time, it is the intention of such a study is the generation of a signal rather than a quantitative assessment. You remember what I said a couple of minutes ago about embryonic stem cells. There we know already what's happening. So there we don't have to generate this signal. We know already what's happening. But in the case of a novel therapy, it is rather the generation of signals rather than being quantitative. And that, of course, has a huge implication for the number of animals to be tested, because a quantitative assessment of a finding may take quite a number of animals, almost like a statistically made study, like a clinical study. And of course, that may actually not be necessary, because you can approach that finding then with more specific tests and maybe including some readouts in the clinical study. And this is exactly what the mindset is. Take a consequence of what happens in the non-clinical study, what you see or what you don't see, and implement a readout, if possible and feasible, in the clinical study. Now, very briefly, at the time, we also invited a few researchers who published either small animal models, like this is a publication just as an example, where scientists established a small animal model for tissue-engineered aortic valve conduits. So obviously these are quite big valve conduits in a small animal, but still that can be done if smartly made. And regulators at the time said, and it's still the case that they're open to consider this. So that was very important, because otherwise it would be a shame to have these developed and the regulator would say we don't look at them because they're not validated or they are too special and we don't want to see them. But we would not go so far to request a large animal study as default. So, for example, for some more tissue engineered products, which are, for example, an artificial organ, you cannot transplant this onto a mouse, obviously. You will have to have a large animal model, but that can become quite tricky. The pig, for example, is sometimes used because of similar anatomy in the abdomen and in the thorax. But to do a GLP study, good laboratory practice in pigs is impossible. So and also it's of course expensive and difficult to do. So we may have to discuss alternative approaches. But of course we wouldn't say no. The regulators wouldn't say no if such a model was employed. So for the duration, we also discussed the duration at the time quite heavily. In some cases, short-term toxicology may actually be more informative, like lifelong follow-up. There were some regulators saying, well, it's a lifelong therapy, a tissue-engineered product, or a cell-based product, or a gene therapy. We need to have lifelong follow-up. But if it is a monkey, that is decades, that might actually be completely unfeasible, or that is completely unfeasible. And obviously, for those of you who do have experience with mice, for example, there are certain mouse strains where there will be spontaneous tumor generation because they are genetically inbred so much that there's almost artifacts coming up later in their lives. So this is then something which has to be put in context with the duration of a non-clinical study. And for dose finding, that is something which is very often very critically discussed in the human study. But here the number of doses may actually be able to be reduced. And then it will have to be put into context with the clinical testing. And for some combined advanced therapies, dose finding may not even be informative or even be relevant. If it's a matrix with cells, then there's no dose. You can of course put fewer cells onto the matrix, but that might just not be informative. And then of course it's again important to distinguish between proof of concept. So what is an efficacious dose in toxicology, and what is the maximum safe dose? And here there are also limitations, because if it's a mouse model that you have chosen, you cannot put an overdose of cells up to a certain content, of course, into mice, because they will just clot up the blood vessel and then obviously produce an embolus, which is an artifact and not a toxicology finding. And this is where we now walk towards a risk-based approach, which is multifactorial. There was actually a group of regulators at the time working on a risk-based approach guideline. It's a quite complex guideline, I must say. And at the time, and also now, I think this is a principle, we should avoid talking about low risk and high risk, because if you call something high risk, it is a bit like a stamp. Nobody wants to touch it anymore because it's a high risk product, but actually it may, after risk mitigation, not at all be high risk anymore. Or after generation of data, it might actually not at all be high risk in reality. So this is why we are avoiding this. But still, it is important to look at the risk and also the clinical context. So for example, genotoxicity, so the toxicity on the germ lines may not be relevant if it is an elderly population, end-stage disease, and so you will have to look at this in the clinical context and the unmet medical need. If this is a breakthrough treatment with extremely interesting efficacy data and so on. Now, this is just very briefly the guideline. I will not go too much into detail here because that is almost an own lecture. Just to say that a risk-based regulation of drugs is not new. Over a decade ago or 15 years ago, we saw the Tegenero monoclonal antibody, the TGN-1412. That is an antibody. It's against anti-CD28, but human immunoglobulins are also antibodies, and they are low risk. This is a very established treatment. So to be an antibody is not necessarily a high risk product. It depends on what the antibodies recognize, and what the clinical setting is, and so on. And that is exactly the type of thinking that we have to employ as well when it comes to non-clinical studies about what we want to look at. And this is a bit of a busy slide, but just to say, this has already come into non-clinical guidelines. So this is, for example, for cell-based medicinal products. That is a guideline from 2008, but it's still the most recent version on the EMA website. And there's a multifactorial approach to risk, as I've just highlighted. So obviously it's the origin of the cells, the manufacturing process, non-cellular components, specific therapeutic use, and then a number of different risk criteria. And that is almost like a checklist where you can look, if you are working on something, what criteria are there, and what criteria can be kind of ticked off, and then you can determine a level of risk and then design the non-clinical study around assessing that risk and maybe confirming or maybe disapproving the particular risk. So as an example, mesenchymal stem cells. These are human mesenchymal stem cells in this case for tissue repair. A very interesting type of cells because they are immunomodulators. So the way they work can be manifold. They can differentiate into certain tissues and repair locally if you put them into damaged tissue, but they might also attract other immune cells or modulate them to the extent that repair is possible by the body's own resources. And you know that stem cells grow exponentially, and they can differentiate in different pathways. So depending on the microenvironment, they could transform into, differentiate into unwanted tissue, or theoretically transform into malignant clones. That at least at some time, and I will come back to this in a little slide in a moment, that was a point of discussion if they are actually tumorigenic in itself. So what toxicology do we expect for a medicine like this? So obviously the behavior at the site of injection, they might de-differentiate and just be fibroblasts or fibrocytes and not do any good tissue at the site of injection, or they might differentiate into something we don't want there, or they might migrate into different into distant sites in the human body and do the same there. And how to test that in a non-clinical model? Now, the first question is, do we have to test that? Because we know already that these are the behaviors that we can see. So maybe we should look at the particular product in case, is it maybe modified? Is it activated cells? Would they do the same? And if so, can we maybe put something into the clinical study rather than see the things that we already know that might happen? And then, of course, the species that we're looking at will have their limits. So cells react species specifically, that's what I said. And cells are immunogenic. So if you inject such human cells into mice, they might actually be attacked, and they might cause immunotoxic effect that you would normally not see in autologous setting in humans, or cells could be eliminated and you might not see anything at all. Interestingly, mesenchymal stem cells can modulate in animals, so it's not always a given that a human cell will not work in animals at all. So there are nice papers out there where inflammatory conditions or other conditions in animals can actually be mitigated by human mesenchymal stem cells, but these are then normally immunocompromised mouse models. And this is exactly the way out. An immunoincompetent animal, then of course you, as said before, you have the advantage that you can test impurities and that you can test the actual product. But of course, then we have to look at the data and think, what do they mean for the human situation? And again, our homologous model that we discussed before, that is also a possibility. So mouse adult stem cells. And there's a lot of data out there, so it may be possible to also rely on certain data out in literature. And that is also a very good approach to do. Just very briefly, there was a publication, it's quite some time ago now, about the risk of tumorigenicity where scientific observations were bridged towards regulatory findings. It's a good read, I would say, if you're interested in mesenchymal stem cells, but that is also one of the principles that there is literature which can be relied upon very often when designing a non-clinical package for the authorities. Now, another example is genetically modified T cells. So T cells are immune cells, as you might know. CD8 positive T cells, they are the so-called cytotoxic T cells or killer T cells. And if you put a tumor-specific T cell receptor, and I'm not talking about CAR T cells here, they might be able or will be able to attack a tumor, but only if the antigen is presented in the right context, so the major histocompatibility complex, MHC, and only if there is a co-stimulation. This is, by the way, where what's the difference with CAR T cells? They have their own co-stimulation with them, so that is not necessary for them. But in a more classical T cell directed approach, you would, and there are products out there which are using that approach, then of course that is different. And the toxicology to expect is one of the findings with the human T cell receptor. So T cell recognition is degenerate, as we call it. So you might have seen mimicries in summer. So this is a wasp on the top, and one of these flies on the bottom, which look like wasps, which children normally get very excited and anxious about, but they are harmless. They use the same pattern, black and yellow, in order to scare off enemies, and that works with children again, as I said. But this is a mimicry, and the same is true for molecular mimicry. This is one of the hypotheses for the pathogenesis of autoimmune diseases. So basically, a T cell does not only recognize what it should recognize, but it also reacts against something which it shouldn't. This is actually something which is life-saving for us, for the immune system, because a T cell is meant to recognize other things. This is one of the reasons, very likely, why the COVID-19 vaccines also work with different mutated strains, and not only the original, because our T cells are degenerate in the recognition, and they can also recognize something which looks kind of like the original strain and can attack it very effectively. And there is a number of older papers, very interesting in the golden age of immunology, so to speak, where you can see that there is quite an overlap of certain human proteins with virus and bacterial proteins. And that is why one of the hypotheses why an autoimmune disease could come up with certain T cells that recognize something. So to cut a long story short, if you have a tumor-specific T cell as a product, they could actually recognize something else. Now, how to test that? Now, if you put this in animals, nothing will happen. It's not relevant at all, because the T cells will not recognize anything, because the mouse does not have the right pointer, the human MHC molecule, which presents that to the T cell. Now, if you take if you would go so far to test an MHC transgenic mouse model, then it would recognize that. But the molecular mimicry obviously may not work, because the T cells may not see what they see in humans, because man is not mouse. So this will have some relevance, but only limited relevance. And the homologous model also has only limited relevance, because it's the same principle. If a mouse T cell recognizes something in mouse and cross-reacts, that might not at all happen in humans. And other way around, if it would not recognize something, it may actually, the human T cell may still recognize that in humans. So what to do? So this is where you would take a brains-on approach, and you would think, where is this antigen actually otherwise expressed? So where would that T cell go outside of the tumor when I inject it into patients? And is there some sequence analogy? There are very good databases out there for T cell antigen recognition, and you can, is exactly what I've shown in previous slides, where you can find these similarities and the probability of recognition. Then also there is experience from similar products. Even knockout mice for that particular tumor antigen may exist. And then you could use additional safety endpoints in clinical trials. So theoretical considerations and also some findings from these analogy, sequence analogy databases. And if you start with a smaller dose, it might actually be safe to progress into humans. Now, before I close the presentation, let's come back to the focus group, because I said that the focus group said, well, can you not publish some of your findings, what you're always telling the companies? And in fact, some colleagues looked at that, and they looked into several dozens of scientific advices with 31 products. Obviously, that is a bit of an older paper, but if you look at current products, it's those issues are still the same, and they very likely always be the same. So there is a lot of knowledge in this. It's open access. So that is the link, and we will put the presentation online, and you can see there is a previous version of this presentation online on the TwinCore website. So you can have a look at that. And just very briefly, very interestingly, there was some questions were almost always asked, or at least very often. So for example, biodistribution was asked in 81% of cases for cell-based products and 72% of gene therapies. Is almost trivial because that is what you would want to know. But tumorigenicity, interestingly enough, was not so much an issue for gene therapies, but more for cell-based products. The reason being that we know quite a bit about the risk of insertion of certain vectors or non-insertion into the human genome. So the tumorigenicity is maybe better described for some gene therapies already than it is for certain cell-based medicine products, which are more individual treatments, you would say. And also very often more than half of the stakeholders and companies asked, can there be a reduction in the non-clinical package? And in fact, what was almost always said was that conventional safety testing was not feasible, and you have now learned why in this presentation. Even a lack of a standalone toxicity study could be acceptable if the proof of concept study actually has some toxicity readouts. And this is, of course, an important information, because if you are planning now a proof of concept study for a research project, then put some toxicity readouts into that study, and you have a perfect study later if you want to develop this principle as a medicine. And also conventional testing for carcinogenicity is generally accepted. So the normal tests, like the AIM tests, that's a mutation test in bacteria where you look if there's if mutagens are present, you can just skip this for cells, because nothing will happen. If you put a human cell suspension into a suspension of bacteria, there won't be any mutations in these bacteria other than there's an impurity in there, of course. So that's normally accepted, and rather a tailored study for tumorigenicity should then be envisaged. And very often also companies asked if good laboratory practice compliance, if it's not possible, is this acceptable? Well, if it's not possible, we can't ask for it, but it must be justified. And GMP is also sometimes not required, but comparability with the later product, which is under GMP, so good manufacturing practice, is important. And normally 3 lots is sufficient to test. But biodistribution and also toxicity should preferably be performed with the clinical study material. And that, of course, may sometimes be challenging, but at least you see the direction of travel of what regulators normally require. So information like this helps you already when you go into early planning. And very interestingly, those colleagues concluded at the time that both regulators and developers must accept limitations inherent in the models available, and that we will have gaps, and that we will have to fill them with complementary approaches. And also the use of more tailored models that are specifically designed that researchers are coming up with will be accepted. So to conclude, this is what I meant with a knowledge and risk-based brains-on approach. So not just ticking the box when designing a non-clinical study, but to do it in a more directed approach. What is it we want to show, and how do we get it best? Can can we apply a risk-based approach to the extent that maybe we do have to do less? It's not always that is risk and even more risk, but it could also be that there's certain risks which are not there, and max out the information you can get. And obviously use the regulatory authority, ask for advice, because regulators will have seen a lot of these different principles and therefore can help. So this closes my presentation. Thank you very much for your attention. And I'm of course open for any question. And if you have a question afterwards, which you thought you should have asked, then don't hesitate to send me an email.
Hello, my name is Rainer Blaschik. Currently, I can't see you, but I would like to ask a question. Thanks very much for this very nice presentation. In a subclause, no, I can see you. In a subclause, you said that pig is not able to be run under GLP conditions. I was a bit surprised about that because pig is such a big model in biomedical research and also in translational research. And if you look at the perspectives in xenotransplantation, I mean, pig is considered the most important donor of xeno organs.
Yeah.
So why is that not GLP compatible, or how did you pronounce it, and why? How is that solved?
Yeah. So this is what my GLP experts told me at the time. They said that it is because of the way that animals are held. It is not possible to have certain cleaning or clean principles that are under the GLP regulations, that you simply can't implement them. But I must say that I'm not an expert in GLP. So if you say that it is possible, then of course that would be a good learning for me. But it is not a problem. This is exactly what I wanted to convey as a message. If you can, if for feasibility reasons and just for the reasons that there are, you can't do something, then a regulator can't ask you to do it because it is impossible to do, or it is so unfeasible to do that you would not have access to a model that, as you correctly say, is a standard model. So that was basically the message that I wanted to convey, and thanks for asking that clarification.
Yeah, but what would be the perfect large animal model? I mean, if we show something in mice, it might not be transferable to humans as if we show that in pigs, because they are much more complex and closer to humans, not only with regard to anatomy, but also with regard to physiology and immunology. What would be then the most perfect large animal model if we don't want to use a non-primate model, which is the most complicated model, also from ethical point of view?
So this is indeed the answer for me. You would, if I had to choose between a perfectly GLP compliant mouse study, which has limited relevance, and non-GLP conformed, but still some principles as you can feasibly do them, conducted pig study, which has a lot of relevance to inform the clinical study, I would choose the pig study very clearly, because that is what you want. You want the scientifically valid information, and of course this is again, you would have to. You would also, when you defend such a study to a regulator, for example, for a clinical study, you would go back to the philosophy of why GLP was introduced. This was introduced in order to give reliability to the data, to the quality of the data that you get. And if you can show that such a study, under the conditions you are performing it, produces reliable data and has been used in other for example, also for other studies, then I think this is perfectly acceptable, because that is why GLP has been introduced. And that's what I meant with brains on. The regulators should not just ask for it because they have to ask for it, but because of a certain reason why they have implemented that in the first place. So that would be my approach when it comes to tailoring such a study towards the use.
Okay, thanks very much. It was good to hear also that regulators are moving away from tick box approaches.
They do. Not all of them, but those who you are dealing with now, I guess. If you would, for example, visit the Paul-Ehrlich Institute in Germany, they are. I think their philosophy is more that they would look at what the feasibility is and what you want to achieve rather than the tick box.
Yeah. Thanks very much.
Thank you. Any other question?
Hi, my name is Anna. Oh. Do you hear me?
Yes. Hi, Anna.
Yeah, perfect. Hi. I have a question. I wanted to know if cell therapy for infection disease like bacterial infection or viral infection is considered in ATMP category or not? Because I have heard from some people that it's not, the therapists, even cell therapists for infection diseases are not ATMP. Is that correct?
Yeah, they are cell therapies. There is an exception for those which are prophylactic, like vaccines. So it depends. It depends on the therapeutic context that you have. So, for example, a vector-based vaccine for the prevention of hepatitis B is not a gene therapy. But to my best knowledge, a therapeutic vaccine for the treatment of hepatitis B, vector-based, would be a gene therapy.
Okay, so it really depends if it is, okay.
It depends. But if you are in doubt, you can, and I would recommend that for a specific product, you can ask either a regulatory authority like the Paul-Ehrlich Institute in Germany, or the European Medicines Agency with a classification procedure, because obviously a lot depends on that classification, if it is a gene therapy or a cell-based therapy or not. Scientifically, you will very likely still have quite similar requirements. So for a vector-based vaccine, you would still have to do biodistribution, for example, and these things. So that's not different to a gene therapy, but certain other things may not be applicable. So it's good to know. But on the other hand, you would not be eligible for certain incentives that only advanced therapies get. So certification of data, for example, that's only available to advanced therapies. So sometimes you can see that companies desperately want to be classified, well, their product to be classified as advanced therapy. And I've also seen cases where sometimes companies desperately do not want to be classified as an advanced therapy. So it depends.
Depends what they want to do.
But at the end of the day, it is up to the classifying regulator, and classification is still a national business. So if the Paul-Ehrlich Institute classifies it as a vaccine or a gene therapy, that's very likely how it is. But if you want to have a European view, then it's the CAT classification procedure.
Okay, perfect. Thanks for your
You're welcome.
describing and answering.
You have to confirm what I said before going into a huge development program, please.
Yeah, sure. Thanks a lot.
You're welcome.